Okay, in this video, we'll cover our last chapter of our course, Chapter 24, Genetics and Genomics. Now, packaged into all of our cells are instructions that are written within our DNA. And these small sequences of DNA are called genes, and they will encode for various particular proteins. And a gene can vary from individual to individual and have different forms. Next, we'll go over two definitions. These terms, genetics and genomics, are used interchangeably, incorrectly used interchangeably, but they do mean two separate things. Genetics is the study of inheritance of characteristics. So what's passed on from your grandparents to your parents to you to your children. That's genetics. And we talk about genomics, that's the study of all genetic information of an organism. So the studying of your genes, what you have, as genomics. So we talk about the whole genetic info from one organism, that's genomics. Talk about how that is passed on from generation to generation, that's genetics. And of all the genome that we have, only about 2% of those genes actually will code for proteins. And those 2% are called exomes. Alright, here's an illustration of a typical human or animal cell, the blue being nucleus. Here's a chromosome. As you pull it apart, you have the histones here, and the DNA wrapped around it. The spiral staircase look to it, and the base pairs. I'm hoping that you've seen something like this in biology or 168. It's basically taking the inner part of a nucleus, finding one chromosome, and pulling it apart where you see these base pairs. And the genes would be one segment of that DNA. Alright, a gene will consist of hundreds of nucleotides and exist in multiple forms called alleles. And for a person that has two identical alleles for a particular gene is described as being homozygous. And a person that's described as having two different alleles for that one gene is called heterozygous. And the way to keep these two terms straight is a heterosexual couple, hetero, is two different sexes, male and female. A homosexual couple is two of the same sex, either two males or two females. So homozygous, two identical alleles, either two dominant alleles or two recessive alleles. And a heterozygous person would be one dominant allele and one recessive allele. So two different alleles for one particular gene. And we talk about where these two different alleles come from. You get one set from your father and the other set from your mother. So this is how traits get passed on from generation to generation. These two terms here are often confused, and they really shouldn't be, because the names tell you what we're talking about. We're talking about the combination of alleles in a person's genome. That's their genotype. So when you're talking about the genes themselves, that's the genotype. Now, the physical condition or health condition that gene sets up or creates is the phenotype. So we talk about the genes, that's the genotype. We talk about the physical condition, hair color, eye color, do you have freckles or not? Do you have an extra finger or not? That's the physical product or physical result of those genes. That physical product is the phenotype. I want to talk about dominant and recessive inheritance. And I'm hoping some of this was or should be review from basic biology or the first part of the course. Dominant alleles will mask the expression of recessive alleles. That's why they're called dominant and recessive. One overpowers the other. The only way you get a recessive trait exhibited is if you have two alleles of that recessive trait. So recessive traits are always going to be homozygous. So two recessive alleles will give you that trait. Autosomal conditions are carried on the non-sex chromosomes. And sex-linked conditions are carried on the sex chromosomes, either the X chromosome or the Y chromosome. That's why they're called sex-linked conditions, because they're found on the sex cells. Okay? And the way you can kind of draw this stuff out, covering the passing on of a condition or passing on of a trait from various generations is called a pedigree. This is a diagram that will depict known genotypes and phenotypes of a family. Here's how a typical one looks. They will vary in what color they're shaded or whatnot, but the shapes always mean the same thing. The squares always going to represent males, and the circles always represent females. And if they're shaded in, or if they're colored, or if they're striped, that means that that person is affected by that condition that you're looking at. So the way you read this is, this man and this woman produce these 3 children here: a male, a son, a daughter, another son. This son married this woman, or produced these 2 children here, these 2 girls. And of everyone on here, this male was affected with whatever disease you're talking about, and this daughter was affected. If they're not colored in, they're not affected. They may be carriers of that trait, but they're not shaded in, they're not affected. Alright, you can lay out a chart of all your chromosomes. This is called a karyotype. We'll talk about some normal karyotypes first, then I'll show you some that are not normal. A normal karyotype has 23 pairs of chromosomes. This gives you the normal pair or normal number of chromosomes that you should have in each cell, which is 46. The first 22 pairs are called the autosomes. They do not determine your sex at all, but the last pair does. Pair number 23 are the sex chromosomes, either XX for female or XY for male. Here's how a typical karyotype looks. Each one has a pair. Remember, these are normal ones at first. So 2, all the way down. These are even numbered to make it easy. The first 22 pairs are the autosomal or autosome chromosomes. They don't impact sex, but the last pair does. These are 2 X chromosomes, so this wouldn't be a normal, at least genetically, a normal female. Compare that to this. Again, they're all numbered to make it easy. Normal, and here you have a X and a Y. There's a very noticeable size difference between the Y and the X, which is what we'll talk about here in a few minutes. So this genetically would be a normal male, because there's 46 chromosomes here. This would be a normal female, 46 and X and X. Now there will be a karyotype on your chapter test. I'm not going to ask you to identify a particular disease, but you need to be able to identify, is this person male or female, and do they have the normal number of chromosomes or not? And if not, what term should be used? Let's see, the somatic cells, or the non-sex cells, will include an X and a Y for males, and of course 2 X's for females. Now all eggs in the female will carry a single X chromosome, but the sperm could carry either an X chromosome or a Y chromosome. So it is the sperm cell that actually will determine what sex that the offspring will be. And sex is determined right at conception, or right at fertilization. If a sperm is bearing the Y chromosome, it will match up the X in the egg and create a male. And if it has, the sperm has a X chromosome, it'll meet up with the X in the egg to create a female. Okay, here's a chart of all this together. Here the egg from the female all have the one single X chromosome. This sperm cell here is bearing an X. So X and X gives you XX or female. And this sperm cell has a Y chromosome, so that Y plus this X gives you XY or male. So it is the sperm cell that determines the sex of the offspring, not the egg. About the genes on the sex chromosomes, like I mentioned before, there's a fairly noticeable size difference between the X and the Y. On the X chromosome, you'll find roughly 1,500 genes. On the Y chromosome, barely 230, roughly 231 protein encoding genes. Now, the terms we talked about before, homozygous, heterozygous, are used when you're talking about a normal pair of chromosomes. You can have a dominant or you can have recessive. But for male chromosomes, they're described as being hemizygous or hemizygous for X-linked traits, because there's only that one copy on the X. There's nothing possible to have on the Y to mask what's on the X chromosome. So there's only one copy there. So you can't have two dominant or two recessive or one dominant, one recessive. Whatever's going to be on that X chromosome, if it's sex-linked, will automatically be passed on because there's nothing there on the Y to counteract that. And some good examples of this are hemophilia and red-green color blindness. These are sex-linked traits that are recessive, and these are always passed on from males or from the mother to their male offspring. And this is much more common in males, both of these. It is possible for females to get them, but much, much more common in males, and they will get this from their mothers. All right, talk about sex determination. Maleness will derive from the Y chromosome, a particular gene called SRY, or sorry, which is kind of funny since most men are sorry. And the sorry gene will actually encode for a particular type of protein called the transcription factor. This transcription factor is what will tell the developing embryo to develop male features and structures and then inhibit the development of female structures. So when this transcription factor is coded for the development of the testes, the penis, the future of secondary sex characteristics as that male hits puberty, and it will prevent breasts from being developed. It will prevent the clitoris from being developed. So this is what will actually determine maleness in an organism. All right, here's a scanning electron microscope image of both the X and Y chromosome. About 1,500 genes here on X, and roughly 231 on the Y. And right in the top part is a sorry gene. That's where the maleness gets determined. The turning on of the development of male features, and then the inhibition of female features. All right, the next three things we'll talk about are very easy to confuse, especially these first two that are here on the slide. And these describe the effects of gender on the phenotype of a condition. And the first one is called sex-limited trait. These are what will affect a structure or a function of a body that's only present in male or females. These are limited by sex. And of course, we're talking about in a normal situation, a normal condition. So, for example, beards, like facial hair, are only found on men. The growth of breasts are only found in females. Those are limited to the sex. Now, yes, these can get impacted by hormonal changes or certain diseases, but in a normal, healthy person, these are limited to either one sex or the other. That's why these are called sex-limited traits. The second one here, sex-influenced inheritance. This is where you have an allele that's dominant in one sex, but actually recessive in the other. And a good example of that is baldness. A male who is heterozygous for baldness will become bald, but a female who is heterozygous for baldness will not be. It is actually recessive in the female, but expressed and dominant in the male. This condition is influenced by the sex of the person. So don't confuse these two. These are often confused with each other. Sex limited, it's limited to one sex or the other, not both, but one or the other. Sex influenced, an allele is demonstrated in one gender but not in the other. And last one here, this is not typically common, it's somewhat rare, but it is worth mentioning. Genomic imprinting. This is the expression of a disorder that is actually determined by which parent passes on that defect. And a good example is what are called Prader-Willi syndrome and Angelman syndrome. These are both caused by exactly the same mutation on exactly the same chromosome. The only difference is what parent passes it on. So if you have a deletion of a very particular section on chromosome number 15, and if it's passed on to you from your father, you'll get Prader-Willi syndrome. If you have that same deletion on the same chromosome, but that defect comes from your mother, that will lead to Angelman syndrome. And these two are totally unrelated, totally different conditions. So exact same defect, exact same chromosome. The only variable here, the only difference is, do you get it from mom or do you get it from dad? If you get it from dad, you get Prader-Willi. If you get it from mom, you get Angelman's. That's what genomic imprinting is. Alright, next we'll talk about some chromosome disorders. Now, any deviations from the normal number that you have can be fairly serious and actually even fatal. The number that you want is 46. That's the normal number of chromosomes in a human body cell. Now, you can have sometimes one less chromosome or one more chromosome or actually whole complete sets. And the number that you want is 46. That number is called the euploid number. The prefix EU or U means true, like eukaryotic cells are true and right cells compared to prokaryotic cells, like in microbiology. But euploid is what you want. That's normal 46. Probably the most dramatic impact of number that you can have on chromosomes is called polyploidy. Poly meaning more than one. This is where you have at least one extra set of chromosomes. So instead of getting 23 from mom, 23 from dad, you get 23 from one and 46 from the other or more. This is going to be a huge impact on the developing fetus, and actually most embryos don't even make it to full term, nowhere near full term. If they do make it to full term, they usually die within the first day or so. There's so many defects there, there's so much wrong, they can't possibly survive. And the way this happens is during meiosis, instead of the sex cell being haploid, having that number of 23 chromosomes, they have 46. There wasn't a complete split of the chromosome. So this is by far the most serious change you can have with chromosomes, having a whole complete set. Next one, aneuploidy. This will include either having a missing chromosome, which is called monosomy, or having an extra chromosome, or trisomy. Mono means one, some or somy means body. So you only have one body when you should have two, one from mom, one from dad. And then trisomy, tri meaning three, you have three bodies. And this occurs during non-disjunction in meiosis. A chromosome pair will fail to separate completely. So when this fertilization happens, you either have 47 chromosomes, which is one more than you should have, or 45. But both of these are considered aneuploidy. Aneuploidy is a generic term that covers both of these conditions, either being monosomic or trisomic. So if you have one less, monosomic. If you have one more, trisomic. Okay, here's how you look at all this information. These two right here are the only normal ones on this chart. So we'll take it back up here. Normal meiosis 1 and 2. These two split here. So half from mom, half from dad. So you get a normal number of chromosomes like you should have. Half from mom, half from dad. So these are normal. But if these don't split correctly, nothing over here and everything over here, everything beyond this point is going to be screwed up. Here you end up with monosomic cells. Here you get trisomic cells. Same thing over here. All because these don't split correctly. Alright, the next few, I'll show you karyotypes of some common diseases and how you can diagnose them just by looking at the karyotype. The first one is Down syndrome. The more formal name is trisomy 21, and it's called that because there are three bodies, trisomy or trisomic, three bodies of chromosome number 21. So if you were to count all these, you'll end up getting 47, but instead of being two bodies here, there are three. 1, 2, 3. So trisomy 21 is Down syndrome. So without knowing anything else about this patient, just by looking at this one image, you can tell that this person will have Downs, and they're going to be male, X and Y. So a male with Down syndrome. Another one called Turner syndrome. Instead of having an X and a Y or XX for female, there's only one X. So this person would be genetically female, but most likely would be infertile because there's not enough female hormones to help sustain a pregnancy. So these are usually infertile. Klinefelters, or XXY, instead of having either XY or XX, you have a combination of both of those, so you have XXY. And even though there are two X's, because there is some Y chromosomes there, this person would be genetically male. And last one, this one for some reason doesn't have a person's name attached to it, like Klinefelters or Turners. It's just called XYY. And like the name tells you, there's an X and two Y's. And this person would be genetically male, because there's at least one Y there. They would probably be a little bit taller than normal, would be a lot more aggressive than a typical male because of the extra male hormones that are there, such as testosterone and so on. And when it comes to the karyotypes that you'll see on the test and on the exam, I'm not going to ask you, is this Klinefelters, is this Turners? I'm going to put up a karyotype, and you'll need to be able to identify, is it male or female? Is it a normal number, the typical euploid number 46, or is something else going on? Is it monosomic or is it trisomic? That kind of thing. But don't worry about trying to identify, is it XYY, or is it Klinefelters, or is it Turners? I wouldn't do that for this type of class. Alright, last thing we'll talk about here, prenatal test. For those of you that have had children, these should not be a surprise for you at all. The whole point of prenatal care is to monitor and screen for any type of potential chromosomal errors or problems that could come up with the developing baby. The first one here, amniocentesis, is the removal of some amniotic fluid to help culture some of the fetal cells. Centesis means to withdraw fluid, and amnio, talk about amniotic fluid. That's where that term comes from. And the last one, CVS, chorionic villus sampling. You are removing cells of the chorion, the structure that would eventually will develop into the placenta, that check for any abnormalities. Alright, that brings us to the end of this chapter, chapter 24, and also the end of the material for the course. And of course, like always, if you have any questions, please ask or post them to the discussion board.